Ok, muy buenas tardes a todos. Muchas gracias por su interés en la presentación del día de hoy. Tengo el placer de tener como invitada a la maestra Beth Fischer de los laboratorios de las montañas rocallosas. Les voy a leer un poco de su currículum para enterarlos de lo que ha hecho ella en su carrera. La maestra Elizabeth Fischer recibió su licenciatura en Biología Molecular Celular y de Desarrollo en la Universidad de Colorado en Boulder en el 83. Posteriormente sirvió en los Peace Corps, que es una organización muy interesante que tiene el gobierno de los Estados Unidos para ayudar en diversas partes del mundo. Y en este sentido ella fue voluntaria en Liberia, en África del Oeste, durante 2 años. Enseñó matemáticas y ciencias en escuelas de nivel preparatoria y secundaria. Después regresó a los Estados Unidos, donde recibió su grado de maestría en biología en la Universidad Estatal de Nueva York, en New Paltz. Durante estos años, la doctora se involucró en cursos intensivos de microscopía electrónica, que es en lo que ella es una experta. Lo verán a partir de su presentación, que tiene unos trabajos increíbles, como se los comenté en la publicidad que pegué en alumnos. Actualmente, la maestra Fischer es la jefa de la unidad de microscopía electrónica y es la jefa de la rama de tecnologías de investigación en los laboratorios de las montañas rocallosas, en la cual provee de técnicas avanzadas relacionadas con la microscopía electrónica para apoyar en el desarrollo y en la obtención de imágenes superestructurales en la división de investigación intramuros, que es donde a mí me tocó que afortunadamente conocerla y que ella colaborara con una investigación que yo realizaba en el laboratorio de la doctora Steve Morton cuando estuve haciendo mi postdoc. Entonces, ella, de las técnicas que ella realiza en este lugar, hace un montón de cosas, pero bueno, ya nos contará ella, hace estudios crioestructurales, hace inmunodetecciones, reconstrucciones tridimensionales por tomografía electrónica, análisis de partículas simples, entre otras cosas. Ha participado en un montón de proyectos que involucran investigaciones con clamidia, coxiela, salmonela. También ha hecho incluso fotografías de organismos mayores, como las garrapatas, que es lo que principalmente se empezó investigándose en los laboratorios de las montañas rocallosas. Ha desarrollado otros proyectos de análisis estructural del efecto de drogas, la falta de nutrimentos, condiciones ambientales, mutaciones, etcétera, con un montón de bacterias que incluyen Mycobacteria, Borrelia, Staphylococcus, Streptococcus y varios virus muy interesantes. En el laboratorio de las montañas rocallosas hay un laboratorio BSL-4. Todavía es el último nivel de bioseguridad que yo conozco. Y ahí trabajaron ébola, virus de la fiebre de Lassa y priones. Entonces, ha hecho bastantes trabajos. Fuera de eso, a la maestra Fischer le interesa mucho aportar a la comunidad, particularmente en el área de Montana, en Hamilton, donde junto con la doctora Kit Tilley y la maestra Anita Mora han desarrollado e implementado programas para llevar a la comunidad. Y en ese me tocó de comentar que viviendo en Montana, mi hijo Antonio y sus amigos, Alton y Cameron, pudieron asistir a estos eventos de lleva a tu hijo a la escuela, al trabajo, y bueno, fue muy interesante porque pues para ellos estar en un laboratorio de este nivel y tener actividades de investigación fue increíble. Además tiene otras actividades en donde llevan justamente estos funciones no es una función, es de mostrar la tecnología en las escuelas secundarias, en las escuelas primarias, cerca de los laboratorios de las montañas rocallosas. Entonces, para mí es de verdad un honor tenerla aquí. A mí me da mucho gusto que haya aceptado dentro de todas las actividades. Y bueno, antes de empezar a hablar en inglés, les comento, les voy a pedir que mantengan su micrófono cerrado. Al final les agradeceré que hagan las preguntas, ya sea en el chat o que abran su micrófono. Si tienen o gustan que yo les ayude con la traducción, con mucho gusto. Y también están dos de mis estudiantes de IFI, para solucionar algunas dudas, Sofía y José. Entonces, sin más, well, I'm going to turn to English. But sorry to keep you waiting and stay in the dark a little bit in Spanish, but I really appreciate you making the time to give us this seminar. And I was telling the students reading your resume and telling them also all the outreach that you do in the community. And for me, it's really a pleasure that you accepted to give this webinar. So the stage is yours. Thank you very much.
Thank you, Dr. Barra. I have to say, I knew Dr. Barra many years ago when he was here as faculty with us at Rocky Mountain Laboratories, and it was really a pleasure knowing him then, and it's really fun to connect now. So I'm happy to be here with all of you. Once I start the show, I won't be able to see you. So if you have a question, maybe Dr. Ibarra will be able to field the questions and you can stop me at any time. You don't have to wait till the very end. So I'll try to keep within time limits. I know your time is precious too. Anyway, so yeah, let's get started.
Okay, thank you. Ya, si tienen alguna duda, dice la maestra que la pueden interrumpir y con gusto yo me involucro en responder. Ok, gracias. Adelante.
Okay, you can see okay?
Yes, it looks
okay,
great.
Great. So I have to, well, maybe let's back up to the beginning again. I don't know about any of you, but for myself, when I was a young girl and when I was a college student and as I was going into my career, I didn't grow up knowing what I wanted to be or do. And it was one of those things. I thought about medicine, I thought about teaching, I thought about all kinds of different things, but really not clear. And I think when I got some more chances to experience electron microscopy, I just fell in love with it. And for me, it's like exploring a whole different world. Whether you're a young child and you look into a glass of water with a straw in it and you see that straw bend, you wonder why. You probably all remember looking through your first magnifying glass, looking at a bug or even your fingers and seeing things so differently. And I have to say, to be able to look at the kinds of things that we can look at that cause infectious disease agents, to see things for the first time that anyone has seen in the world and try to understand it in the spatial context has been really a fun journey for me and something I hope I can share with you, the excitement that it brings. So I wear a hat as an electron microscopist, but I also am part of what's called the Research Technologies branch. And what this really is a group of different people that have gone into science but fell in love with the technology that is applied to scientific questions. So we have a lot of different scientists involved with these technologies. It becomes project specific. And really, as Dr. Barr and I were talking before we started, it's a way to be economical because a lot of these technologies are very expensive. So it makes sense to have people really specialized with them that can come and utilize them well and apply them to everyone. So we're a core facility that provides support for a lot of different people. In my particular group, we supply the imaging tools used for both confocal and light microscopy, as well as electron microscopy, and that's where we're going to spend our time today talking. But other technologies that we have in our branch include things like flow cytometry, genomics, protein chemistry, structural biology, and our visual and medical arts. I think I heard Dr. Ibarra mention Anita Mora, and she colorizes a lot of our images, but that's the group that does that. So they're very specialized in more that communication piece of bringing things to life for publications and out in the media. We also have what's called the integrated data science section. All of these disciplines require a strong bioinformatics component, and so we built an ecosystem where informatics can apply to a lot of these different areas. Where are we? Well, we're up in Montana, as Dr. Ibarra said, and that's pretty far away from where the main NIH headquarters is, which is out in Bethesda, Maryland. We're about 2,300 miles away. The population of the entire state of Montana, which encompasses all of this region, only has about 1 million people. And I know in Mexico City, you have close to probably between 8 and 9 million people. So you can imagine this is a pretty large area and not a lot of people. In fact, we have a lot more cows than people. Compare that to the distance from Hamilton, Montana, where we are, to Mexico City. It's actually almost the same distance. You're at about 2,500 miles away. So that gives you a little perspective on how far away we are from our mother program. The main campus in Bethesda, Maryland has a lot of buildings. It houses not only the National Institute of Allergy and Infectious Disease, but 26 other institutes and centers with about 18,000 employees. And this is the big building 10 clinical center. This is where a lot of that translational research to a product that can be used in the clinical setting or bench to bedside, as we like to say, can happen. A lot of very experimental, but also therapeutic vaccines and things like that. People are trying, people are desperate. Back here in Montana, we're completely different. We have a small little campus here that has a nice river flowing through our valley and beautiful mountains. We have a 33-acre campus, about 450 employees compared to the 18,000 back in Bethesda. We're only NIAID, so only part of the National Institute of Allergy and Infectious Disease. And we have quite a few buildings, and it's been growing as well. As Dr. Ibarra, I think I heard mention of some of these. Some of the things that we study in the bacterial realm include one of the diseases that actually was the reason why our lab is here, and that's a whole interesting lecture in itself. That's Rocky Mountain spotted fever. Other things that have been studied here include Borrelia, Francisella, Coxiella, chlamydia, prions, and neuronal viruses. About 15 years ago, a level 4 lab went in with, and it's under the guidance of Dr. Heinz Feldman, who runs the laboratory of virology. And that's where we study the level 4 viruses, but also some level 3 and 2 viruses that are produced that can give us understanding of the level 4 mechanisms. This also included the area that we really started our SARS-CoV-2 discussions and research. So now I'm going to switch and really focus on, pun intended, I guess, on the microscopy and the resolution revolution that's occurred over the last 15 to 20 years. And to really help you appreciate how far microscopy has come in such a short amount of time, I'm going to take you way back. So we're going to go back to about the 1600s. And there was actually a lens maker named Zacharias Janssen, and he's actually credited with building the first microscope and the first telescope, although there's some controversy about that. But he developed what's called the first compound microscope. So he actually put two lenses together, and that's what made this different than just people looking through a spectacle. And this gave him the ability to really start seeing things a little bit differently. And you can see these small little handheld ones. It actually became not for scientific research, but really sort of just for fun. People would go around and they called them flea glasses. They could just look at objects, but it was more of a novelty than anything used for science. A little bit later in 1659, Antony van Leeuwenhoek, who's also from the Netherlands, started actually looking at things and categorizing things. So he started looking at animals. He's the one that's first thought to have looked at single cells and really started to use it in a more scientific way. And he came up with this quote, which I really liked. In all falling rain carried from gutters into water butts, animalcules are to be found. So he discovered that bacteria, that there were small living things that our eyes couldn't see. And he called these animalcules. And that in any and all kinds of water standing in the open air, animalcules can turn up, for these animalcules can be carried over by the wind along with bits of dust floating in the air. So really, he's the first one that described really bacteria. And you can see it in these drawings he made back in the 1600s. Then along came Robert Hooke, and he became very systematic. He was from England, and he really started using it more scientifically. He started looking at the cork of bark and noticed that they kind of looked, it reminded him of the cells that might be in a monastery where monks reside. So that's where the term cell actually came from. And so he was considered sort of the father of microbiology. Through the 1800s, the evolution of the microscopes didn't change very much. They sort of changed in look and appearance, but they really didn't change a lot in what they were able to do. Light sources were added, mirrors were added to direct the light sources. But then along in the late 1800s, a German physicist that worked in a Zeiss factory for microscopes named Ernst Abbe came up with an observation. And what he realized was there was a law where the resolution of an object or our ability to distinguish it was relative to the wavelength divided by n over the sine of alpha of that wavelength. So it's a little bit hard to understand what this means, and I'll try and explain it to you. But think of it this way. If this is your wavelength, which is kind of long, this object here is going to appear as just one big blob. You're not able to see it as this wave can't resolve it. Whereas if you have a smaller wavelength, now you have that ability to distinguish this as one object and this as another object. And this was a really critical point in time for the microscopists and how that would change the way things were designed. So he really came up with some lenses to cut aberrations. And during this time, Rocky Mountain Lab started to become into existence too, a little bit later. So in 1906 in our lab, this area was known for its high mortality rate for the agent that causes Rocky Mountain spotted fever. And so a physician came out from Chicago named Howard Ricketts, and he came to study the origin of this. And people thought maybe it was from the water. This was a highly big agricultural area, and there was a lot of growth. So, of course, people with real estate were nervous if it was the water, nobody would move here, and there would never be land development. But he spent some time and he actually figured out that it was ticks that transmitted the agent of spotted fever. And again, that's a whole nother history that's quite fascinating. Later, there was another doctor here, Dr. Cooley, and he was a entomologist who studied the life cycle of the spotted fever ticks. So he used microscopes heavily in his research. Dr. Cox of Coxiella fame found he could grow rickettsia and chicken eggs and allowed mass production. Again, he used microscopes. And then we had Dr. Ralph Parker and his wife in 1916, who spent their scientific honeymoon studying ticks under the microscope as well, trying to characterize the different ticks and their feeding parts and where these bacteria might reside. Now, if I were looking for a husband back in 1916, that would have been my guy. Okay, so along came the physicists, and they're the ones that really made some important observations that led to the development of an electron microscope. In 1900, Max Planck, who many of you have heard of, I'm sure, described that energy exists in discrete quanta or discrete object. And Albert Einstein then postulated a couple of years later that photons exist as particles or discrete quanta. And then Louis de Broglie came up with the wave mechanic theory that stated that these photons would travel in the shape of a wave. So if you put all these together, Ernst Ruska was able to come up with the notion that if he used a much smaller wavelength, so we talked about wavelength and resolution, if we use the energy of an electron instead of a photon, we'd be able to resolve or see things much more clearly. And that's because the wavelength of an electron is about 1,000 times smaller than the wavelength of the farthest visible light that you can see. So in 1933, he came up with a design and was able to build an electron microscope. Instead of glass lenses or lenses made out of glass-like material, he used electromagnetic coils, and that was able to direct the electrons down a column. But essentially, the idea of an electron microscope and a light microscope and the way they operate is quite similar. You have an objective lens, you have a projector lens, you have a source of illumination, which is in this case is electrons instead of a light source. And you also, of course, have a way to record those images, which prior to the electronic and digital camera era was on film. So the electron microscopy at Rocky Mountain Labs also started a long time ago. Back in 1953, Dr. Edgar Reeby was shown here looking at one of the early microscopes that was first commercially manufactured in the globe. And we had that here at RML, and they looked at things like Mycobacterium tuberculosis, tobacco mosaic virus. And here you can see, you know, I think most of us would understand Mycobacterium or the TB bacteria don't look like little flat sacks like these are pictured. But the fact that they were able to image them at such high quality was really phenomenal. A lot of what came later was how to prepare samples a little bit better. And you're going to see how in the last 15 to 20 years, that's really revolutionized what we can do today. So currently in our microscope facility here at Rocky Mountain Labs, we provide both 2D and high resolution 3D imaging. We have expanded cryo-EM capabilities both in Montana and Maryland, and we're going to talk a lot more about that. And then we also have light microscopy support here at RML as well as in Maryland. So we do a lot of different kinds, and we're going to kind of go through all these different kinds of microscopy. So you'll have a great understanding by the end of this of what we can do. I have a great staff, and I always want to bring them up first because, of course, I'm just sharing some of the stuff that they've all collected. And so I want to acknowledge I've got an outstanding group that have been here for a while. Some Dr. Varl will remember, like Vinod Nair. We have a lot of newer members. Brian Hansen's been here for quite some time. He started actually as an intern when he was going through college, as did Forrest Hoyt. They're both permanent staff members now. Dr. Jacqueline Lung runs our light microscopy facility. These are some of the interns we've had, and this is another employee that we have here now, Sophia. Dan Bruno operates our spatial technologies. I'll talk about that briefly at the very end. I think one thing to also keep in note that we aren't in a vacuum. We have a lot of thanks to all our leadership that we give support through, all our collaborators, a lot of administrative help, and then we have a lot of overlap with our other sections. And I think as you see towards the end, we're not in a vacuum by ourselves, that we work closely with our light microscopy facility, the biological imaging section. We work with our structural biology group. We now also work with our genomics research section, flow cytometry, and protein chemistry. So some of the light microscopy capabilities we have here, we have a Zeiss LSM 880 with a very scan fast and an NLO multi-photon detector that allows us to look into tissues a little bit more deeply. We also have a slide scanner and then an inverted scope for light, and it's a dedicated image workstation that goes with that. And I just want to point out here, and this is kind of why the expertise is important. Anyone can be trained to take a picture of a sample, but it all starts with a good sample, but also knowing how to optimize parameters on a microscope or any other technologies that you're using. So we had a postdoc that's just down the hall from us here. In pre-consultation, these were the kinds of images that he was generating. After he spent time with the core facility and got some training, that same sample now he could image at much higher resolution. So again, that talking of resolution, where here you see kind of green blobs and spots, here you see them in great detail. And these are rickettsia that are growing in cells. They have these beautiful actin tails that you're able to see polymerizing, which helps with the motility of the bacteria. So again, anyone can take a picture, but to take a picture that offers meaning and really drives the technology to its greatest extent takes some background training and knowledge. So here's what some of the microscopes look like. This is the confocal. This is the high throughput slide scanner. The slide scanner can do great things. You can set it up to montage. So you can take a picture that has 1021 fields of view, 3 different color channels. It's a pretty large file, or large tissue piece, 38 millimeters by about 20 millimeters. But that allows you to get high resolution where you can zoom in on a piece and actually still be able to see it in high quality and not have it full of pixels. And again, looking through deep things, this is salivary glands from a tick, but being able to distinguish different parts of the gland and what's going on in there is kind of important in these studies. So again, getting that resolution is important. And then she's also expanding our intravital imaging capabilities. We're having set up here this spring something called the Leica Stellaris Dive. This is going to allow animals a better look into animals, looking deeper into their tissues, being able to use a multi-photon component. We can look at live mice. You can track things like a brain, for instance, follow neurons. So especially if you're looking at viruses that might affect neurons, you're able to track that over time. Jacqueline has also been developing new ways to look at things. So along with the technologies comes a lot of different techniques that we try to develop. So she's doing something called tissue clearing. If you try to look through a microscope at a tick, you're not going to see a whole lot. So how to preserve it, but make it somewhat transparent so you can see what's happening was the point of this particular thing, which she has been able to achieve with Dr. Adam Nock. And then, of course, you can look at other things too, mouse brain, skin tissue. We're also getting something called a light sleep fluorescent microscopy, which I don't have time to talk about today, but it again provides higher resolution that's much gentler on the tissue, so it allows longer imaging. And then beyond that, if you are into the light microscopy side of things, the big buzzword years ago became going from confocal to now the super resolution of something called STED. And that along with expansion microscopy, where you actually expand the tissue, this almost looks like an electron microscopy picture, but it's actually taken with a light microscope. Again, this has to do with the way you can prepare a sample and then special techniques that you can use on a microscope to image. Now pause just for a second there, because that's the world of light microscopy, and see if anyone has a question, or we can wait to the end too.
As you wish, Beth. I'm not sure. ¿Tienen alguna pregunta al momento o quieren esperar al final? Pero si gustan hacer alguna pregunta ahorita. Hey, Beth. So I'm really surprised about the. Of course, I always knew that you guys have a beautiful work over there. But I'm surprised how you guys are trying to clear the ticks. So are you guys giving us what do you do with the keratin and all that in ticks to look inside them?
Yeah, it's quite a protocol, and I can, if you guys are interested in that, I'd be happy to put you in touch with Jacqueline. But she goes through a lot of exchanges in solvents and really starts extracting out some of those pigments, but at the same time trying to preserve the integrity of the location of the tissue, so you're not disrupting that. Now, if I were to look at that by electron microscopy, I'd say that was disrupted a lot. but but by light microscopy, I think it does allow for you to visualize things that are more or less intact. So if you're trying to track a bacteria in a tick and kind of see over time where it is, as opposed to having to rip out the midguts or rip out the different, parts and smush them and look at them, this provides a way for being able to look in vivo, which can be a little more insightful than just trying to figure out where things came from after you smash them on a slide.
Yeah, that's true. And then the other one, the stat, that's really interesting. So, I mean, I'm just impressed. I guess that will take another seminar, but
It will, and I think that's the exciting thing. It's, a lot of these technologies are starting to approach, well, in the case of light microscopy, I think they thought they'd reached the limits of resolution, but people have figured out different ways to manipulate the optics or computationally to drive that resolution even further. So it's, yeah, you're right. That could be a whole lecture in itself. It's really exciting time to be in the imaging side of things, as all technologies. I mean, we're all benefiting from that, whether you're in science or other things too. The world is very technology driven.
yeah, you're right. I mean, that's one thing that I mentioned my students in my class, that they are living in an incredible moment, same as us, like all the revolutions that the nanotechnology, computers, and all that. It's just amazing. How,
Yeah, well, fasten your seatbelts now, because I'm going to show you some basic stuff, but then we're going to get into the really fun stuff that we get to do as electron microscopists that rely a lot on computer skills for sure.
great. Well, thank you.
Okay.
Okay.
Yeah. Okay. So for our EM core facility here at Rocky Mountain Labs, we have um, what's called a Titan Krios. And that lets us see sort of, well, let me start back here. It lets us see things under cryo conditions. We have our scanning electron microscope where we can scan the surface of things. We have our 2 transmission electron microscopes that allow us to sort of see general things, which we'll talk about a little bit too. And then we have something called a focused ion beam, which we'll also get into. So with your conventional 2D imaging, here's a picture that Dr. Ibarra will recognize, salmonella with the beautiful flagella. So I think that's a picture maybe you were asking for even a little bit ago. We can take, so this is a whole mount of bacteria. We can also take tissues or cells and be able to cross-section them, and we can look at those in more detail. So, we look at cells that are uninfected, but then we look at things that are affected. So we can try to understand, how is a bacteria or virus getting in a cell? What compartments are they in? And we'll talk more about that as we go forward. And then things like this Lassa fever virus entering a cell. There's a lot of different mechanisms. So transmission EM kind of gives us a lot of basic information about what's happening at the cellular level. With the focused ion beam technology, I'll show you a little bit more how this works too, but basically you have an electron beam that does the imaging and an ion beam that starts cutting away the material of the sample in increments of about 4 to 10 nanometer slices. So you can imagine if I cut this away, take a picture, cut it away, take a picture, I can rebuild that into a 3D volume. And we'll talk about this a little bit more. This is in a rabbit lung tissue, and these are tuberculosis bacteria and a granuloma. We can also do what's called freeze fracturing of a cell. This is a HeLa cell. And one thing we'll talk about a little bit is why we want to freeze stuff instead of just chemically fixing things. But basically, it keeps things in more native state. So here with the HeLa cell, you see the nucleus, and then fractured along these planes, you can see Golgi apparatus, mitochondria, various vacuoles, endoplasmic reticulum. Lots of the cell machinery that needs to function properly for us or that needs to be harnessed by a pathogen to take over things. And then we'll get to the cryo-EM as well. And when you look at chlamydia, when it's not in its frozen state, these membranes get all jammed together. You don't see them, but when you're looking here, you actually see individual protein membranes poking out of the cell. We can look at viruses. We can look at interactions of viruses with a Fab antibodies, so these would be vaccine targets. We can look at prion fibrils and go to high resolution with those. We can look at antigens from things like the flu virus or the anthrax toxin. We work on a lot of different things. So basically, we can encompass a wide range of current imaging capabilities from both the atomic level of things up through light microscopy that's actually starting to even border with that clearing technique whole organisms like a tick. So again, our 2D imaging capabilities include the TEM, so transmission electron microscopy, where we transmit the electrons through the sample, the scanning electron microscopy that allows for surface imaging of things. This is a tick mouth part going through a feeding membrane. Tuberculosis bacteria, again, these are prions, freeze fracture. And then something that's important, which is immune labeling, which I'll talk about why that's important shortly. Where we can label different parts of the cell. So we see bacteria in there, but we want to know what proteins are they expressing in that space, or what proteins is the host cell expressing in things that might come in contact with the bacteria or virus. And then we can do something called correlative light and electron microscopy, combining the techniques where we can fluorescently label something that might be transient or hard to find in the electron microscope, and then find that same cell and be able to identify those structures and correlate them. So they can be colorized, and as Antonio said, these can be colorized by our visual medical arts group. These are used a lot in the media. And I'm going to try, I think I'm going to be running out of time, so I'm going to try and speed up a little bit. So as I mentioned, immune labeling is something that's really critical for us to be able to identify proteins that might be interacting with a pathogen. And there's the main players here include the antigen of interest. So this could be the SARS-CoV-2 spike protein, or anything sticking on a cell that gives us a protein that we're interested in, an antibody of some sort against it, and then something we can see in a microscope. So either fluorescent tag in a light microscope, or something electron dense like gold, or some kind of dark reagent. And then the section that it resides on, or the sample itself. So if you have specific labeling, you see a beautiful gold bead that's very specific on a particular antigen. If you have background labeling, you're going to see gold everywhere. So not every technique is good, and it doesn't always apply. And there's a lot of different ways to perform it. So it's not as simple as following a recipe. It's understanding what you're looking at and where things might have artifact. When it works well, though, you can label things. Like in this particular case, we're labeling Golgi apparatus, and this is looking at rickettsia inside cells and trying to figure out what it's doing to the trans Golgi. We can use things like fluoronanogold, where you can see it both by fluorescence as well as in the electron microscope. Now, the limitations of 2D imaging, of course, are that you can't always get what you think in 3D. And so what appeared to be a hand, in fact, was not. And this is something we have to remember. We're really looking at the shadows of things when we transmit electrons through a sample. And so a lot has happened in this field to try to come around that. And part of it was pushed by these three gentlemen who won the Nobel Prize in Chemistry back in 2017 for developing cryo-electron microscopy for high-resolution structure determination of biomolecules in solution. They were able to figure out a way to freeze a sample so it kept its native state. It didn't have the impact of having a lot of chemicals treating it to preserve it, and then be able to look at it in an electron microscope. So why freeze? When we're freezing things, this would be our native state, say, of peas. If we chemically fix them, they may not taste like peas anymore. But if we freeze them, it actually retains their structure a little better. They're not quite as raisin or wrinkled up. So it gives us a more native state ability to see things. And so we're able to do that with our Titan Krios. We have two of them here now. And this allows for us to study things like ion channels and membrane surfaces. This is RNA and the way it's folding, down to high resolution. And the way this works is we take a sample, we freeze it in a grid in suspension, and it's kind of like a hole or bubble wand where you are able to take that picture in a suspended particle, and each picture is actually made up of a movie. So each movie can then be realigned into a clear corrected micrograph. So it's a series of images of the same spot. We now get a clearer view of this particular virus, and then we can pick the particles out of the field of view. In this case, we're picking a protein, and you can start assembling these proteins with like particles in the way they look the same, that the way they are oriented in the ice is similar, and they kind of categorize in that way. Then you can back project those and start building a model that's going to show you where those shadows came from. And as you feed that model back in and collect a lot more images, like thousands more images, in some cases 100,000 or even 1 million images, you start to develop a really high resolution model. So now when you're looking at things like a vaccine target and where it binds, this is really important information. And we'll give you an example of that in a little bit. So back to the prions. So we're looking at the prions here. This is what it would look like in the raw micrograph. These are the 2D classes that came from those fibrils when you computationally average those together. You can dock in crystal structures from the sequence, if you know the sequence, and start folding it. And where this is really important, and you think about a prion, we can't have necessarily an antibody against a prion because a prion protein is something that's natural in your body. This is just a misfolded version of it that can cause things like mad cow disease. And so for these kinds of misfolded proteins, it's really important to think about structure-based drug design. And to do that, you have to understand the structure and then where you might find targets for different kinds of drugs that can interfere with that. We can also do something called cryo-electron tomography. In this case, we take something that's maybe not symmetrical, like a whole bacteria or a virus that doesn't have symmetry. We can flash freeze that at minus 180 degrees Celsius. Again, you're on that grid and you have a frozen sample. In this case, instead of taking lots of pictures of the same things that are oriented in different ways, we're taking one field of view at very low dose and taking pictures of it at a tilt. So you can think of it like an MRI of your knee, where you, instead of tilting the machine around your knee, in this case, we're tilting the sample and taking pictures from a lot of different angles. Those can then be back projected into a model also that tells you what shadow angle that came from, how it's going to generate into a 3D volume. So once you have that, you're able to rebuild it. You can slice through it, much as you do in a confocal microscope, and really start seeing things at atomic resolution. So this has allowed us to study things like, in this case, VLPs. This is influenza particle with HA and NA spike proteins. These are the things that change in flu every season. That gives you the different H1N1, H5N1. It's a hemagglutinin and neuraminidase. In this particular study, we studied once they enter a cell, you can look at how the membrane curvature, depending on cholesterol content, can change, and how that changes its ability to actually enter the cell. We can also use something, as I mentioned, called the dual beam scanning electron microscope. So this is a focused ion beam that erodes away at the sample, and then the electron beam that comes back and takes an image of the tissue that's remaining or the cell that's remaining. So this allows us, besides tomographies, you can only do very thin samples. This allows us the ability to go through an entire cell. And so in this particular case, you can now reconstruct things a little bit more deeply. And these are kind of all the modalities I just showed you all on one page. So let me give you an example of how we apply some of these technologies in the study of SARS-CoV-2 on viro cells. So RML received notification that there was a pneumonia of unknown cause that started in China that started to alarm people. Within about a week and a half, the laboratory of virology, operated by Heinz Feldman out here, came up with a plan of attack on how they would start handling this. The first SARS-CoV-2 sequence came online, and from that, they developed a spike protein, made custom antibodies. By January of that same year, at the end of January, they discovered it was the ACE2 receptor on the surface of cells that was the target for this virus. It's incredible to me, this timeline, because it's so incredibly fast, really in the first 3 months of a virus coming on, where all of a sudden you had the isolate received here at RML, we took pictures, you had the known receptor, you had antibodies against it, studies for what treatments might be available began, animal studies started to happen, and that's really just in the first 3 months of the pandemic that all of you have lived through. So why is that important? It's important to figure out where it's binding so that you can figure out ways to stop it from binding. And that's one way to treat this, as opposed to after you've already been sick with it. So of course, that's where you start building interest in a vaccine design. So in this particular case, Barney Graham from our Vaccine Research Center, that's part of our institute back in Bethesda, took the sequence and, along with Jason McClellan down at Texas, solved the structure for the spike protein of SARS-CoV-2 based on the sequence. So they were able to build the sequence, come up with a 3D structure, and that allowed for a vaccine target really rapidly. And in particular, the messenger RNA vaccine came out of this study, the mRNA. And this is where they took a bit of the viral genome and packaged it up. And that's what is used to be injected in bodies to basically give us the ability to produce this target spike protein, which then our body will generate a lot of antibodies against. And that allows us, of course, to fight the virus before it can take over and make us too sick. Dr. Emmy DeWitt spoke on May 6th on a lot of her animal studies. I'm just going to briefly go through some of them. All of you have access to what's called the videocast.nih.gov. And if you look up Emmy DeWitt on May 6th of 2020, you can actually see her lecture on this, which is quite good. And there's a lot of other lectures too, not just on SARS-CoV-2, but it's a good site to go to if you're interested in infectious disease stuff. So again, early on in the pandemic, they were able to use rhesus macaque monkeys. They inoculated the monkeys with SARS-CoV-2, and they found within just a few days that inside a lung tissue, there was this complete pneumonia happening. And so you can see all this infiltrate of immune cells, and you can imagine the lung is usually more like a sponge where you can breathe quite easily. When your lung starts looking like this, it's not so easy to breathe. So we also looked at lung tissue at 7 days post-infection, trying to find evidence of the virus. We saw some damaged cells, and they were able to also find some viruses lining the airways of these cells. And again, you have all this would normally be empty space in your lungs, but it's just filled with a lot of fluid and edema. We are able to use antibody labeling to study the disruption of Golgi and how it traffics SARS-CoV-2 through a cell. So we can look at 1 hour post-infection of a cell. You start getting these small changes that are blown up here. Here's your Golgi. You start getting these little singular membranes. By 3 hours, you get more of these little vesicles forming, which start to become quite prominent by 6 hours. By 24 hours, you have a lot of double membrane. These are the factories inside the cell where the virus is generated. And you can see how they've really taken over the cell in this picture. So again, looking at these different compartments in different ways, we're able to identify areas of possible intervention. So we could do focused ion beam. This is where we use that ion beam milling to go through an entire cell. You see all these little bubbles are where the SARS-CoV-2 virus is forming. All the blue on the outside is virus on the surface of the cell. Here in the cutaway, you'll see green in mitochondria. The pink is the nucleus of the cell. And in yellow is that giant replication complex where all the virus assembly is occurring. And here's the cutaway of that as well. So it really gives you an appreciation for, one, how large this viral complex is, but also how the mitochondria seem to have some kind of, we don't know what, but I think some groups are starting to study it. What are the mitochondria doing, and how are the viral particles harnessing that energy from the mitochondria, because they seem to recruit it around this giant replication complex. We can also use that correlative light and electron microscopy to look at small events by light microscopy and then co-locate them and find them by electron microscopy. I'm going to skip this picture and jump ahead to what we did for the SARS-CoV-2. So here we looked at trans-Golgi network, strep, the calnexin, strep, and MERS. So this is kind of our control, but the calnexin and the TGM show us the endoplasmic reticulum and the trans-Golgi network involved in this. These are transfected cells, so they're not always in every cell. They sometimes can be hard to find. So Forrest and my group was able to locate these small cells that were of interest and then find out what structures were associated with particular proteins, whether it was a spike protein or the envelope protein, non-structural proteins, a membrane protein, or open reading frame 6 protein, and see what effect they had by themselves on the cell without the whole virus being there. And so why is that important? Well, you've got the viral particle that has those different components. It gets in the cell, and they're responsible for different elements of this binding. So if you're able to figure out where that particular protein is being successful, it gives you, again, an idea of where you might be able to interfere with that and have that chemotherapeutic intervention. So we're going to step away, and this is, I think, about my last slide. I think I have one more. But as I said at the beginning, one thing that's interesting about the technologies is how, instead of it just being electron microscopy over here and light microscopy over there and genomics over here and mass spec over there, all these technologies are starting to converge, and the way we look at things start to converge, which gives us a lot of functional information along with spatial context. So we've just started a new technology in the last year or so that's called digital spatial profiling. We're using both nanostring and Visium platforms at this point. But this is really a combination of microscopy and genomics. So we can take formalin-fixed, paraffin-embedded, or cryo sections, and we can get protein or RNA readouts. So basically, you can identify regions of interest. This is, again, a SARS-CoV-2 infected mouse lung. But you can find a region of interest and then map particular proteins and know where it came from. So if you're looking at what immune cells are being activated, and not just what immune cells, but what protein in the immune cells are actually being activated, this gives us that ability to localize that or find out the genomic information in a particular region. So with that, I'm going to end and just say the light microscope opened the first gate to the microcosm in this quote by Ernst Ruska. The electron microscope opened the second gate, but what will we find in the opening of the third gate? And that's the exciting thing for me. The technology fields move so fast, as you all know, and the impact it has on the science is incredible. Ernst Ruska did earn the Nobel Prize, or was awarded the Nobel Prize in 1985 for his 1933 invention of the electron microscope, and luckily he was still alive to enjoy that. So muchas gracias, and I'm happy to take any questions.
Thank you very much, Beth. That was really beautiful pictures. As always, it's just amazing the work you guys do, and it's just impressive. So while you were talking, Lisbeth placed a question, so I'm gonna read it to you. It says, I would like to ask if these types of incredible images can be consulted on any website, or if there is a repository, or if they are only obtained by researchers. So they're mainly that, yeah.
Well, some of our pictures you can find on the NIAID Flickr site.
Okay.
So anything publicly available, I mean, of course, in publications too, but if you type in NIAID Flickr, which I'll put in the chat,
Huh?
you'll be able to pull up many of the images that I showed today, in addition to a whole lot of other ones.
Okay, great, thank you.
And they're publicly available, so if you want to use them, they're free to use for presentations too.
Yeah, thank you. As a matter of fact, I don't have it right now here. I'm not sure if I ever told you, but here in our school, we prepare manuals for the lab classes. And interestingly enough, the one they use for the one of the classes is general microbiology. It has a picture, yeah, that you took for Olivia on salmonella. So it's the salmonella with the ruffles.
Oh yeah, I know the picture you're talking about.
Color. I think the ruffles look yellow and the bacteria it's orange.
Kind of a
That
pinkish
one.
color. Yeah,
So
that you'll find that. I think you'll find that one on the Flickr site.
yeah, so basically that one, it's for me it was very interesting because getting to know you and Olivia, of course, and then finding that they use that picture here at my school is really interesting. So it's kind
Yeah, oh, that's very flattering. Thank you for sharing that. I think what's really great is that the pictures are always worth 1,000 words. And you know, we took the first pictures of SARS-CoV-2 that were used in the world, and I think people, a lot of people wrote to us and to ask us about them, but it really speaks to how powerful an image can be. And of course, there's the artist or the artistic part of it too, right? I mean, a lot of these organisms are incredibly beautiful, and we're really fortunate to be able to see them and visualize them and then share them.
And I'm very fortunate to get to know you because then, like I tried to get you during the pandemic, but I knew you were really busy. You guys never stopped. So, but I'm really happy that this time you were able to be here. So thank you very much. There's another question by, sorry, they're coming, so give me a second. So how does cryomicroscopy affect the observation of infectious agents, especially in terms of sample preservation? I guess if there is an effect, if the cryomicroscopy affects sample, I guess that might be.
Yeah, so that's the power of the cryo-electron microscopy. We're able to freeze it, so we haven't put a lot of chemicals on it. So it really is more native state. With the microscope itself, we have to be cautious because if you hit it with a lot of electrons, you start to have radiation damage. And that's why the pictures themselves don't look that interesting. It's sort of like, okay, there's a little fuzzy spot in that ice, and she's telling me this is high resolution. But I think what's amazing is the information is in the image. And that's where, when you use the computational power that we have and the algorithms to solve that, it becomes really interesting to be able to put that together and make sense of it. So yeah, it's been a game changer when you're trying to figure out where exactly what amino acid is this antibody binding to. We're able to figure that out now, and we're working a lot of different studies for a lot of different, drug design things, but also antibody things that impact a lot of infectious disease. So the impact it has on the infectious disease world has been tremendous. Not only infectious disease, though, in material science too, they use the same kind of microscopes, and they can actually watch chemical reactions occurring at the atomic level. So it's something that's amazing for biologists, but in the material science world, it's also quite interesting and has had major impact as well.
Oh, that's great. So thank you. There's another question by Bren. Congratulations for all your great work. It's really amazing. Here is my question. Where else can we use structural profiling? I mean, can it be used in a community of organisms like a biofilm or just tissues?
Oh, that's a great question. Yeah. So thank you, first of all. And I think that it can be used with other things. There's, depending on the technology you use, there's limitations on what probes are available. So unless you have a lot of money to custom make different probes, you're limited a little bit right now to human things um, and mouse and non-human primates. I think dogs are also coming out now. You can label things. So if you know you have a bacteria over here, you can label that, of course, for fluorescence. But if you want to know immune cells that are coming into it, that could be interesting. Biofilm-wise, I think that would be a little tricky. And again, it depends on your question. There might be other techniques that can be applied, again, through either correlative techniques, through laser microdissection techniques. There's a lot of different technologies that might be helpful if it's outside of those organisms. We've done like blood smears and like bone marrow and things. So it's not tissue necessarily, it can be a cell spread, but then you sometimes lose that spatial context as well.
Okay, great, thank you. There's another question by Daniel. How much time does it take to obtain a cryo-EM image?
Now that's a great question too. So the images themselves are pretty quick. Right now on our microscope, it's highly automated, so we can collect about 400 images per hour. And each of those images, again, is a movie. So one single image is really a collection of 50 frames. So think of it as a high-speed camera. We're shooting lots of frames, and then we can realign those. But yeah, on that microscope, about 400 per hour. On our scanning electron microscope, it takes about 2 minutes to collect an image.
Wow. And then you also need a lot of capability for storing all those images.
We generate terabytes of data per week. So yes. So how to manage all that data? I mean, again, it's easy to be, okay, I can freeze a sample and take a picture, but all the computational power, high performance computing it requires, the storage of terabytes and terabytes of data is all, that's the whole IT world that also factors into scientific discovery.
Wow, that's impressive. It's like all the rest of the, I mean, I'm not sure you can call this part of the omics. I guess it's not. It's a different, but it's benefit also for all these new technologies. Like in genomics, you get also a lot of information. So, but luckily we have now more storage everywhere. So that's really great too.
Yeah, and actually our genomics facility here used to be the big data producer. We overshadowed them a long time ago with our cryo-EM.
I'm impressed
But yeah.
because I remember when I was there, I think Steve is not anymore there, but I remember his unit used to generate all that with chemo and all these people. But now I'm really impressed that you guys overwhelmed them. So there's a couple of other questions. One is by Orlando. Is it possible to visualize protein DNA interactions and the residues implicated in that specific interaction?
Yeah, we actually have been doing some of that, and it's quite interesting. We're kind of limited on size, so about the smallest size we can go down to is about 65 kilodalton complexes. Anything smaller than that's a problem. But when you have a DNA complex, especially with a protein, that actually bulks it up pretty well. And sometimes we'll use tricks like we'll put in, we have a protein that's pretty stable. We can put another tag on that so we can bulk it up a little bit to be able to visualize it. But a lot of our proteins are around 85 to, 500 kilodaltons in size. But once you get really below like 35 kilodaltons, then you're really talking X-ray crystallography.
Yeah, how pure do you need the proteins to do that?
The purer, the better.
But sometimes, there's always these soccer proteins that come together with your proteins, so it's hard to get like 90.
Yeah, and that's where we don't work in a silo, right? Like if you were to ask me how do I do that, I would say, I don't know, I'm not a chemist. So I'll send you over to another one of our technology sections where they do the structural biology, and they're biophysicists. And they'll say, oh, we can help you with that. We'll figure out how to stabilize, how to purify. So it's again, not being in a silo with the way you collaborate with people is really important, right? You have to share information. You have to draw on each other's expertise and strengths. And then you can figure out solutions to these problems.
That's true. And I kind of picture which is your beloved series this season? Because you mentioned a lot Silo. I'm thinking you like that series. Did you?
Yeah.
Yeah, I like it too. So I haven't finished it yet, but I will. And there's so many series now. But I picture, like you mentioned Silo, and it's like, I think Beth might like that series. So I better finish it. I don't spoil it for me. So here's another question. And I guess this is for a kind of IT question, but it says by Ruai, what kind of computing power is needed to perform that kind of microscopy? I guess the one that generates so much data.
Yeah, so we have multiple computer systems. So we have computers that operate the cameras. When the data is collected, it comes into basically a server environment that's on a computer that's just a halfway house that can house several terabytes. But that quickly gets put over to a high performance computing system, and it uses a lot of GPUs and CPUs. And that's as far as I can give you. But we have a lot of all of that, and it is pretty intensive. Yeah. There's
Yeah,
a lot of great programs available to help with that. We can kind of watch the data coming off live now with something called CryoSpark Live, where the data is coming off, and then we start getting information on, is this going to be a good data collection? Are there a lot of particles? What resolution are we starting to get already? And that's within seconds off the microscope. So yeah, that computational power. And anyone interested, if you're ever in Montana, please come visit. We're always happy to share and show you stuff for sure.
that's another good invitation. If you ever go over there, that's definitely a place you want to stop. But is the security to get in still that difficult, or?
I just need your passport, right?
Oh, okay, okay, and then we'll call you. And the good thing for everybody listening this is that after that there's the brewery, which adds another spot that I really love. The Bitterroot Brewery. It's a really good place to gather after work. So
Yes, come in the summer, then the daylight hours are long.
yeah, that's true. ¿Alguna otra pregunta que tengan? Pueden hablar, abrir su micrófono o adelante con las preguntas escritas. So I have a question. I'm glad I was able to. I'm really happy that the students are asking. But in the meantime, I kind of like your the scientific honeymoon. I didn't know about that fact with Parker. That's really interesting. So he took his wife over there to
I
get.
know she had to be quite a woman.
Yeah, now we know a lot of stories, like a lot of women that previously they were acknowledged. They play a really important role in their husband's life, and sadly for some of them they were not recognized. But now we know their stories. Like I didn't know about Esther Lederberg's story, and it's very interesting how after Joshua Lederberg, she divorced, she still continued doing a lot of stuff, and her story is just really nice. And getting to know, maybe who knows? I mean, I'm sure she was working with him, and she was also interested in visiting the Bitterroot Valley for her.
Yeah, I don't know the story behind her, but it's interesting to think about it. Oh, I see one pop up. What do you think is the third door of microscopy?
There you go.
You know what would be amazing? Everything we do in electron microscopy is frozen or chemically fixed, and it's a snapshot in time. And so we don't have that time frame of what's happening, what came first or what came second or what caused what always in place. We can combine that with light microscopy and live cell imaging to go so far. But wow, would it be powerful to see some live microscopy by electron microscopy? See how these molecules really interact as you see a protein antibody coming up against a virus and how it's pushing that aside and how it's docking in there. We're doing a lot of small molecule drug inhibitors right now that go and, really small things that go in and take a protein and change it. So all of a sudden it doesn't work anymore. And just to see that in action, that would be cool.
He says maybe like attosecond pulses.
Okay, you're charged with that.
Really good. Okay, ¿alguna otra pregunta? This is really nice. I'm really happy that you were able to do it, and I'm gonna take your word, and I might ask you for Bob's contact. So I'm really happy. Like we have had other persons in this seminar, but I think the techniques that you tell us is just taking us to sometimes even for the kids, it's not come to the next level and they don't even know, because sometimes I don't know, we just start with our curricula and there's so many new things coming up. So there's another question that just popped up by Elizabeth. Would you like to read it or would you want me to?
Oh,
I will,
okay.
I would like to express most sincere gratitude for sharing your invaluable knowledge during the conference. The presentation was inspiring and gave us a unique perspective on microscopy. I think that's really nice. Thank you.
Oh, thank you very much. Yeah, I think like I said at the beginning, you can look through a magnifying glass and you have no idea when you're 4 years old or 5 years old that'll lead you to an electron microscope. But I think that's the thing. You find things that are interesting to you and the ability to be able to explore that. And as Dr. Ibarra just said, not even knowing what's out there is a big part of it. So it's just being open to discovery and seeing what's out there that makes science actually really fun.
No, thank you very much, Beth. And given that like I told you before, I have a visitor from Hamilton. I'm gonna send you a small diploma that we prepare for you. Let me show it to you. But you're gonna have it in print because I asked Carlton to give it to you. Can you see it?
I can, thank
Oh
you.
yeah, I know it's in Spanish, but still it's something for thanking you for giving us this seminar. And I look forward the next time I visit Montana, I can stop and go to that brewery. And I need to tell you, I mean, we drove, my wife and I, we drove from Mexico City all the way to Missoula.
Wow.
So this past summer, that's a crazy thing to do, but we enjoyed it a lot. But we couldn't stay only 3 days with my son in Missoula, so we couldn't really stop in Hamilton for that long. But hopefully next time we go and visit you and Bob. How about that?
Yeah, that would be great. And we'd love to see some of you here too. So any of those students, I think, there's always fellowship internships as postbacks or postdocs. So as you go on in your careers, keep us in mind.
Yeah, please. Will you guys open your cameras? ¿Quieren abrir sus cámaras? So we're not only watching black numbers, there you go. So these guys are my students. There are a lot are gathered. There's one.
Oh, wonderful.
This is my lab and these are my students, and then the rest is also more students. Si quieren abrir su cámara y quieren salir en video, lo trataré de subir a YouTube. La liga de la asistencia ya la pegué. And yeah, there's more people opening. So thank you very much to all, and to you, Beth, for sharing with us all these very interesting things. I knew it was going to be a mind-blowing thing.
Thank you for the invitation and really nice to see all of you. Good luck with your studies and come visit.
Yeah, sure. We'll try to do that. You see, guys, hay una invitación para ir a Montana y como dijo la maestra Beth, si les interesa, ahí siempre están. Es un lugar muy bonito. Montana es un lugar muy aislado, pero al mismo tiempo alguien decía por ahí, pueden ordeñar una vaca en la mañana y pueden ir a trabajar en el laboratorio en la tarde. Entonces, es un lugar muy interesante, con mucha ciencia que se hace, con muchos recursos, afortunadamente para ellos. Y afortunadamente es dinero público y todos esos son recursos que podemos también como extranjeros aprovechar. So, well, thank you very much. Gracias a todos. Thank you very much, Beth. And,
Muchas gracias.
muchas gracias, Beth. We'll see you soon, I hope. And please
I hope so.
please say hello to Vinod, Brian, Olivia, Anita. I still have nice memories of that place. I really enjoyed my stay over there. So
I will.
I know
Until next time.
yeah, thank you.
You're
Gracias a
welcome.
todos. Gracias, muchas gracias. Gracias a la maestra Beth. Gracias a todos por asistir a esta presentación. Y en este momento está la liga puesta ahí. Por favor, tienen 10 minutos para que yo la cierre. Y gracias a todos. Thank you, Beth.
Goodbye.
Bye. Adiós a todos. Gracias.